Which of the following are fused in somatic hybridization involving two varieties of plants? Which of the following are fused in somatic hybridization process? So pollens callus somatic embryos means these is the product formed. So this is the product formed after fusion. So somatic embryos also the answer will be protoplast. So what is protoplast? Cytoplasm with nucleus is known as protoplasm. So the protoplasm of two different cells will be fused to form somatic hybrids. The process is called somatic hybridization. So the answer will be 3. Next. So here the answer will be protoplast. Why the answer will not be pollen? Pollens are haploid cells. So in this somatic hybridization, we require fusion of two diploid cells. Only then a haploid cell will be formed. This parent cells will be taken from two different species. Then after the process of hybridization, a hybrid will be formed. That is somatic hybridization. So we need a diploid cell. Diploid cell for somatic hybridization. So pollens are haploids. So that is not possible. Callus matlab from callus itself we will get different varieties of cells, right? From callus we will get root cells, stem cells, leaf cells. So it will be undesirable if we select the callus for hybridization. We will not get the desired hybrids. So callus will not be the answer. Somatic embryos means somatic embryos, they come from a single somatic cell. Single somatic cell undergoes division and becomes an embryo. There is no fusion taking place in this. So somatic embryos will not be the answer. The answer will be protoplast. The two different species, two different cells taken from two different species, and the two protoplast is taken and they are fused to form a diploid hybrid. This is called somatic hybridization. The answer will be C. Next, match the following: citric acid cycle, glycolysis, CTS, proton gradient. Glycolysis occurs in cytoplasm. So only with the help of this we can cross these options. So the right answer will be maximum this. फिर भी हम चेक करते हैं. Citric acid cycle occurs in matrix of mitochondria. Then ETS, electron transport chain occurs in inner mitochondrial membrane. See, this is the mitochondria, right? ETS occurs in inner mitochondrial membrane of mitochondria. Here is the double membrane mitochondria. This is the intermembrane space. This is the matrix. And this is the inner mitochondrial membrane where the electron transport chain or electron transport system is present, like this. Electron transport system is present. Proton gradient proton gradient occurs in intermembrane space of mitochondria. So in mitochondria, see, I'll show this here. This is the mitochondria, double layered. Okay, electron transport chain occurs here, and electron transport chain occurs here. This is the space, and this is the inner membrane. Proton gradient after synthesizing protons, they enter into the intermembrane space of mitochondria, and there will be a lot of accumulation of protons in the space. So again they need to go see, this is the outer membrane, this is the inner membrane. Okay, the F0 F1 system will be present here. So due to accumulation of protons in the matrix, there occurs a gradient. And due to accumulation of protons in the space in inner mitochondrial intermembrane space, there occurs a gradient between matrix and space. So the protons must move from space into the matrix by synthesizing ATP. So the proton gradient, we will see the proton gradient in intermembrane space. The answer will be second option. Outer membrane, inner membrane. On this inner membrane, there will be electron transport system. From the matrix, first protons will move to the intermembrane space through inner membrane is permeable for protons, right? So through inner membrane, protons move from matrix to space, and again in space there will be more protons get accumulated, and then the protons will move from space to matrix with the help of this electron transport system by synthesizing ATP. For every 3 protons entering from space into the matrix, an ATP will be synthesized. So more concentration gradient of protons will be seen in space of mitochondria, intermembrane space of mitochondria. The answer will be second option. Which of the following statement is correct regarding the process of replication of E. coli? So E. coli is a prokaryote. Read the question properly. Replication in prokaryotes. They are asking replication in prokaryotes, not eukaryotes. So in prokaryotes, during replication, the enzyme used was DNA polymerase, whereas in eukaryotes, the enzyme used was RNA polymerase. So we need to see for DNA polymerase. Here RNA polymerase is there, so strike it off. And during replication, the replication process, that is the polymerization process, takes place in 5' to 3' direction only, because 3' direction 3' end contains free OH group, which is helpful for the formation of bond with the next upcoming nucleotide. Right, with the next upcoming nucleotide there should be some bond formation, and this free hydrogen bond, free OH bond helps, free OH group helps in formation of bond. And so 5' to 3' should be there for polymerization. So this is 3' to 5', strike it off. This is also 3' to 5'. Right. So what is the option left? DNA dependent DNA polymerase catalyzes polymerization in 5' to 3' direction. So So replication in E. coli occurs in 5' to 3' direction. Why it is called DNA dependent DNA polymerase? Because from a single DNA we are getting another DNA with the help of DNA polymerase enzyme. So this process is DNA dependent DNA polymerase. In eukaryotes we can say DNA dependent what RNA polymerase. This is in eukaryotes and this is in prokaryotes. Okay.